Well, good afternoon, everyone. Right, and the last point that we were discussing was about identifying the major sequence from precursor hairpins. Right. So, if there is capital R in the nomenclature, that would mean that it is a major sequence. While if there is a small r in the name of the sequence, that means it is a precursor sequence. Okay. Moreover, the database accession number were also modified in order to differentiate at accession level itself whether the sequence is major sequence or precursor sequence, right? So if the accession number itself contain MAT that I have kind of highlighted over here, that would indicate that it is a major sequence, while the accession number of the sequence, if it don't contain this MAT, it can be considered as precursor sequence. So 2006 May, one of the major feature that included was that they initiated the genomic context to this micro RNAs. Okay, so they started to give provision of coordinates of the genomic position of each miRNA sequence, like where they are present on chromosome, what is their location in genome, in what chromosome they are present, where they are present, so on and so forth. Okay, so these were the major feature of 2006 release. Okay, moreover, 2006 release was one of the most important, update in terms of target detection. Okay, so what happened was that these guys also devised an algorithm in which target for the given miRNA was being able to be predicted using this service. Okay, so for detecting target or predicting the target, these people have devised an algorithm called as Miranda algorithm. MI there stand for microRNA. Okay, Miranda algorithm for target prediction. So it was quite a simple approach for identifying the target for given miRNA sequence. It involves mainly of two steps. The first step being detecting the potential binding site, and in this step, potential binding site would be predicted based on complementarity. More is the degree of complementarity, higher are the chances that the given miRNA would go and bind to the mRNA sequence. That was the main premise of the first step of detecting potential binding site. The second step that they carried out was to filter out those sites that do not appear to be conserved in multiple species. Right? If they figured out during the first step, okay, we have got complementarity of the given RNA in the given mRNA, then they would compare that complementarity with different species as such. Okay, and if the same region are found to be conserved, then it would be considered as a target prediction. So one of the reason behind doing this comparison over multiple species is the premise that this miRNA functionality is supposed to be conserved throughout evolution, right? As we have discussed already, this miRNA functionality is conserved throughout evolution. So unless and until it is conserved, you cannot consider it as potential target. That was the premise behind the second step. Okay, so due to this second step of comparing the sequence of whatever has been identified as potential binding site with multiple sequence has got far-reaching effect. So the biggest advantage of using this approach was that it worked out very well for species that have clear defined orthologs in closely related species. Like, well, so for example, the miRNA target sequence would be very similar in case of human, mouse, and rat. Why? Because all of these are mammals and all of these are primates, and therefore, in due course of evolution, less amount of mutation might have been accumulated in that particular group, right? But the biggest advantages started to accumulate when the accuracy, as you consider, alignment or comparison with distantly related sequences, we'll say, for example, from plants or prokaryotes, or we'll say, for example, distantly related clades like mammals or some sort of like aves or reptiles. These species have uh, diverged a long back ago. Therefore, in due course of time, their mRNA sequence, their target sequence might also have accumulated mutation. Therefore, detecting such distant homologs had become difficult in clearly identifying the target. So that was its one of the biggest disadvantage. As the conservation becomes poorer, when we include some diverse sequences, the accuracy of detecting the target would eventually decrease. That is quite logical. So of course, there are some plus points, there are some minus points of using this Miranda algorithm. So the point is that in 2006 version, target detection utility was integrated in this database, and it is still present over there. You can check it out. Okay, in 2008 version, what they have developed is that 2008 ke aate, NGS sequencing came into existence and popularity. Therefore, genomics based tools were also incorporated in this case. Okay, so more sequences were added. They incorporated genomic tools to distribute comprehensive miRNA genomic information. Okay, in 2008, clustered miRNAs were also dealt with, as I have already mentioned in my previous lecture, that the precursor can be monocistronic or polycistronic. But in previous version, there was no utility of taking care of these clusters of miRNAs. Okay, so in 2008 version, provided a list of clustered miRNA if there are any. Okay, so a new search facilities was also developed that would allow the user to retrieve clusters of miRNA in any organism, and people were also made, people were also enabled to search the clusters with, if they are separated by a certain distance. So such type of query building was enabled in this version. Okay. Moreover, other genomic features, like if there are some CPG islands associated with microRNA, or if there are transcription start sites near or around it, or do it contain EST or cDNA overlap. Okay, so we'll say, for example, if it has got sorry if it has got transcription factor binding site nearby, or if there is polyadenylation site prediction, poly A site prediction, if it is there. So the thing is, all these features were eventually added in 2008, right? So here is how the features would look like. We'll say, for example, if this is the miRNA that you are interested in, then in its surrounding region, what are the different genomic features that are available. So it might help people to properly gain a kind of global understanding on how this miRNA would function. That might be one of the logic behind implementing these genomic features in this. Okay, so the later versions, like the version that is published in 2011 and 2014, well, they incorporated the deep sequencing data to the resource. Okay, so in this deep sequencing data, what they did is that they mapped the read from short RNA deep sequencing experiment to the microRNAs in MirBase or MirBase, right? So in order to make this data accessible, they developed a new web interface to view these mappings. The data was also associated with given microRNA annotation if there is any. Filter reads by experimental and counts were made available. search for microRNAs by tissue and stage specific expression was also enabled these uh, uh, in these revisions. Okay, and the latest database now contains some 24,500 microRNA loci from say 206 species. Okay, so it has kind of grown in last 10 years or so. Well, now I am not aware if after 2014 there is any significant increase or there is any modification in this MirBase. We would have to check out this NAR issue. If there are any, I would let you know. Okay, so this is for you for your further readings. Well, these are all the papers that has described your MIR base through 2004 till 2014. Okay, so I would advise you to go through all these papers and explore what things have been done, how things have been done. Okay, and I would also demonstrate you a little bit of how do you roam around in this MirBase during our demonstration section using this website. But if you are curious enough, maybe you can visit this particular website and start out exploring for yourself about nomenclature, about genomic location, about that Miranda algorithm. Just explore these things. Try to do queries and everything. Okay, so this is all about your MirBase. Okay, the next resource that we are going to, we are going to deal with today is something called as LNC-RNA-DB. Okay, so maybe I would stop this meeting for time being, and we would begin our discussion on LNC-RNA-DB in 5 minutes. Okay? Okay, beta.
Excuse me, sir.
Yes.
हाँ बोलो.
So Vaishnavi Malviya here. Sir, I joined late. My screen kept freezing.
Okay. Who is this?
Vaishnavi Malviya.
Okay, ठीक है बेटा.
Sir, Shipra also.
Shipra.
Shipra full.
Okay.
Sir Shivani
Sir,
Singh.
Pooja also.
Pooja also, right?
Sir Shivani Singh also.
Okay.
Sir Garima also. Sir, I actually text you also.
Okay. Yes, I have seen that. Namrata? Namrata is still not here, right? Okay, ठीक है बेटा, कोई दिक्कत नहीं.